introduction is utterly unnecessary. For those of you who are not, I cannot imagine a better guide to the subject matter. Professor Gilbert is Emeritus Professor of Biology at Northern College and Finland Distinguished Professor at the University of Helsinki, trained in molecular biology, developmental immunology, as well as in history of biology. He is the author of one of the most translated handbook of developmental biology, now at the 12th edition. He has been one of the pioneers of evolutionary developmental biology, evolution, and is actually the major supporter of its extension to eco-evolution, merging embryology, medical physiology, ecology, and evolution. To whose agenda, he particularly contributed with the revision of the notion of organisms in terms of holobionts. Throughout his innumerable papers and books, which earned him several awards, he is committed to the use of history as an exercise of critical thinking in science and a tool for promoting a more correct and responsible communication between science and society. How the turtle formed its shell reflects the lifelong focus of his evaluable research on the issue of evolutionary novelty as applied at one of its most fascinating and debated case studies. I'm very grateful to Professor Gilbert for accepting our invitation and we will come.
Thank you, Sylvia, and thank you all members of the Stazione for inviting me. As I mentioned in an earlier lecture, this has been incredible for me to be here. I'm sorry I can't address you in Italian. My grandparents were not biologically Italian, they were culturally Italian. They spoke about their trips to Italy. My grandfather worked for Fiat in Torino, and for my grandmother, the center of the universe was La Scala. And who's to show her wrong? Yeah, as a matter of fact, there's a lot of interesting family history that Italy relates to. But I'm going to be talking today about how the turtle gets its shell. How does nature make novelty? How does nature create newness without destroying that which has worked before? And what I like in this, whoops, okay, back again, maybe. And how does the turtle get its shell? This is from a book for 5-year-olds. It was sent to me by Karen Crawford, and when I got it, I go, oh shit, we've been scooped. But how the turtle gets its shell is a good 5-year-old's question. And these are the questions which are critical in biology, and often the ones that we adults embarrassingly don't know the answer to. It's also a good question for the marine laboratory, and this is a picture of Anton Dorn and Ernst Haeckel from a paper by Christian Gopin. And here's Dorn over here, and Haeckel. And one of Dorn's big contributions was this notion of evolutionary development or evolutionary change through change in function. And that's what I'm going to be talking about in the turtle today. I'm going to be talking that its ribs have changed their function. The ribs of a turtle no longer support the rib cage. They no longer are involved in the thoracic cavity of the turtle. They've gone outward and they are making the shell. I probably will not have time today to talk about the plastron, which is a wholly different set of bones. We have some theories on it. Maybe I'll be able to express it, or maybe I'll just say, invite me back again. Okay. So, Karl-Ernst von Baer said, but the developmental scheme in the case of turtles must be modified in a quite unique fashion, which on closer examination of their development makes us very interested. Unfortunately, he couldn't convince many other people of that. He convinced one particular person who really did a good job on it, but basically, the turtle was a very hard organism to study developmental biology in. And one of the reasons is that it is the most non-model system ever. If you tell your students you want a model system, you're telling them you don't want the turtle. Hardly anybody works on it, and that's for good reason. It takes about 6 years for them to reach sexual maturity. You're not going to do QTL analysis on the turtle. They're only available 6 weeks out of the year. Fortunately, these are the weeks when my students are also available. They have a clutch size of about 10 to 20 eggs a year. They don't get thousands. You know, they're not sea urchins. They're not fish. You get maybe 10 eggs a year from each clutch. There's only one mutation known, and that's albinism, and it's not napped. The eggs are very fragile. They are very difficult to grow, and they are not transparent at all. So they're really bad. The advantages of working on the turtle is if you want to work in a laboratory where you don't fear competition from big labs. This is important to me because I work with undergraduates. I don't have graduate students until the last 5 years of my research. So yeah, I change projects when other people come into the domain. Also, anything you find will be new and potentially publishable. There is a lot of low-hanging fruit. We were the first people to look at molecular basis of turtle development. It's great for undergrads and getting funding for science education. You'd never get funding for this for a graduate student. But I could say, I'm teaching people how to do developmental bio. That I could get funding for. And third, everyone loves turtles. Best public relations in the world. So one gets photos on the covers of journals more frequently than the merits of the publication. People are pissed off. I could have gotten my picture on the cover of Cell, but you guys published a 2-page review article and you've got turtles on the cover. That happens. Okay, turtles are also arguments for creationism. And this is because of the paleontological fossil record is very poor in turtles. That says here, Creation Museum in Kentucky, turtles appear abruptly in the fossil record. This is a problem not easily explainable by the evolutionist, but it is easily explained in the biblical creation model. Since turtles appear abruptly in the fossil record, that seems to be what they do. The only thing turtles ever did quickly. The current data are consistent with a creation event followed by considerable diversification coupled with degeneration. We support belief in an intelligent designer, the God of the Bible. Okay, so turtles are, the fact that we can't tell how turtles evolved is being used by creationists to say, well, that's because they didn't evolve. It sometimes gets personal. This is an article, I wrote an article called Turtles at Loggerheads. I wrote it with Tyler Lyson. The title of the paper by Lyson and Gilbert summed up the evolutionary conflict perfectly. Turtles all the way down, loggerheads at the root of the Chelonian tree. This sentence from the closing paragraph is just as candid. The new discovery of the beautifully preserved Odontocalus fossils produces more questions than it answers, reopening questions of turtle origins, shell evolution, and original paleoecology. You can find the correct answer based on true history to the question of the origin of turtles and the timing of fossilization in the Bible. Turtles were created on day 5 of Creation Week, only 6,000 years ago, and this fossil dates from the flood of Noah's day, approximately 4,500 years ago, a hugely violent worldwide event. Okay, so they get personal here, and I have to go and find out what's happening. Okay. So, I've been told that I should go through, for some people, about Evo, Devo in general, and then kind of go into what's happening with turtles. Usually when we talk about evolution, we talk about natural selection, the mechanism being descent with modification. There's variation in a population, look around you. There's competition for survival, most animals dying before reproducing. Survival of those animals most fit for their environment, and the offspring are from the survivors. And modern synthesis says the offspring inherit the genes that made their parents fit for their environment. This is the 5-step method that we teach in our classes for the Darwin-Wallace notion of natural selection. Huxley, Darwin's friend, however, said evolution is not a speculation but a fact, and it takes place by epigenesis. He didn't say it takes place by natural selection. He's looking at a different level. It takes place by epigenesis, by body construction. And he contended that a theory of body construction must complement the theory of change, which is natural selection. he wasn't so much interested in the survival of the fittest. What Huxley was interested in was the arrival of the fittest. Huxley did embryology. But until a century later, we did not have a good theory of body construction. And Huxley writes to Darwin, "It is true that the more widely two animals differ from one another, the earlier does their embryonic resemblance cease. But you should remember that the differentiation that takes place is the result not so much of new parts as of the modification of parts already existing and common to both of the divergent types." This is the frontispiece, The Man's Place in Nature, which kind of emphasizes there's really nothing new being created here. It's the modification of what's old. Okay, so Evo Devo really goes from Huxley's view. We're looking not about the survival of the fittest, but the arrival of the fittest, that evolution results from inherited changes in development, and that a more comprehensive theory of evolution results when the population model of evolutionary change, based on allele frequency, is supplemented by a developmental genetic change of body construction based on gene expression. So how does gene expression give new phenotypes? And fortunately, there are probably only 4 ways that this is done. But before I get to them, I'll just mention this wonderful quote by Hughes and Kaufman on the modification of arthropod bodies. "To answer this question of arthropod adaptations by invoking natural selection is correct, but not sufficient. The fangs of a centipede and the claws of a lobster accord these organisms a fitness advantage. However, the crux of the mystery is this: from what developmental genetic changes did these novelties arise in the first place? How do you get something new? By changing the embryo." And I'll just go over here. There are basically 4 ways of tinkering, and here's Mother Nature with an isopod here. You can change the time of gene expression, okay? You could change how long a gene is active, for instance. You could change the place of gene expression. Put gene expression in a new cell. The expression of a particular gene is going to be in a new cell type. Change the amount of gene expression. It's the same cell types making the same gene, but at higher amounts. Or you could actually change the kind of gene being expressed. And when we talk about the genes, we usually talk about paracrine factors, transcription factors, and signal transduction pathways. This is kind of the center of developmental biology at the moment, where a paracrine factor is a compound, usually a protein, made by one cell, which is recognized by a receptor on a neighboring cell. So here's the cell here, here's the extracellular, here's the cell membrane, and there is a receptor for a paracrine factor. It's a specific receptor just for certain paracrine factors. And the paracrine factor is released from one cell type. It is now bound by another cell type. When the paracrine factor binds to its receptor, it changes the shape of the receptors inside the cell, giving them a new activity. Usually this activity is a kinase activity. It can phosphorylate proteins. So here it phosphorylates itself, and now it phosphorylates other proteins, and there's often a cascade of phosphorylations where the cells are not enzymes until they have a phosphate on them. Once the enzyme puts on a phosphate, they can become new enzymes, and they eventually phosphorylate or change a transcription factor, allowing the transcription factor to enter the nucleus. And when the transcription factor enters the nucleus, it binds to the chromatin and changes gene expression. So you can get from something on the outside of the cell, a protein made by a neighboring cell, to change gene expression inside the cell. This is really the culmination of biology in the 20th century in animal development. The notion of paracrine factors, transcription factors, and the signal transduction pathways that link them. So when we ask, for instance, how does the webbed foot of the duck evolve? How do you get a difference so that the duck has a webbed foot, but the chicken doesn't, you can look at the place of gene expression. So here's a chicken foot, adult chicken foot. And when we look to see how this chicken foot got to be 3 digits and not have a web, what one finds is we start off with a web. We usually have webs. We, as embryos, have webs, but we have apoptosis in those regions which will die. These cells in here will die in the web. And what causes that apoptosis? Well, BMP is a protein which does many things, but if it's in the webbing of the foot, it will cause these cells to die. And it was thought that, okay, this will do it, because once you have BMPs, that will cause the webbing to disintegrate. And sure enough, it does. So you start off with webbing, and then the webbing goes away. Now this is looking at gremlin protein. Gremlin is an inhibitor of BMPs, and it doesn't exist in the chicken foot. So BMP can do its thing and tell the cells to undergo apoptosis, which leads to the freestanding digits. In ducks, however, they have their webbing. They have a very limited amount of cell death, but they also have BMPs in their webbing, but they have lots of gremlin in their webbing, and the gremlin will stop the BMPs from working. You can actually put gremlin into a chicken foot, chicken limb, and get a duck type of limb out of it. You can change the timing of gene expression, and we see this in the human brain, where the human brain keeps the fetal growth rate for about 2 or 3 years after birth. And there's all these genes which are candidates for hypomorphosis. Since the human chimpanzee split, the loss of GAD45G, the loss of SRGAP2, which are growth inhibitors, positive selection for ASPM and microcephalin, so that the cell division genes are kept on for longer periods of time. The amount of gene expression can be changed. And here we have Darwin's finches. And the amount of BMP4, which in the facial mesenchyme causes cell growth and mitosis, it causes proliferation, what you find is that BMP4 is correlated with the broad beaks, whereas a loss of BMP4 is correlated with the narrow beaks. So here we have the finches that crack nuts. Here we have the finches that go into the cacti to get insects. Very different habitats or very different resource use, and lots of evolutionary studies by the Grants on these finches. But the proximal cause, how do you get the variation? It's BMP4 expression in the craniofacial mesoderm. Then, as I mentioned before, you can actually change the gene involved, and that's responsible for the insects having only 6 limbs, hexapods only having 6 limbs, because there is a mutation which gives a polyalanine region of the ultrabithorax region. And ultrabithorax is expressed in the thorax and abdomen, and what it can do here is the polyalanine region represses the gene for distalis. And distalis is a gene that initiates leg development. And so basically in insects, but not in other arthropods, think of the seafood salads, lots of legs in shrimp and lobsters and crabs. Okay, so we now have these 4 ways of tinkering, and we can get to the turtles. Okay, but I tell the biochemists that there's a large subunit and a small subunit of the turtle. We work with Traphemis scripta, the red ear slider turtle. In America, it's sometimes called the Woolworth turtle. This is the turtle that you could get for $0.25 at the department stores. And the carapace is on top, and what we're seeing here are the scutes of the carapace and the plastron on the ventral side. And today we'll be talking mostly about the carapace. Okay. And here are the bones underneath those scutes. This is of Geochelone, the Galapagos tortoise. And what we'll see is that actually the ventral skeleton, the plastron, is formed in very different ways than the carapace. So we have something new. We have a turtle shell. So how does the turtle form its carapace? What makes a turtle are its ribs. Okay? The turtle ribs enter the dermis of the back skin rather than forming a rib cage. So here's a turtle seen from below. You're kind of at, its digestive tube looking upwards. And here are the vertebrae of the turtle. And you can see the ribs coming out of each vertebra. And then you see the ribs going and extending all the way to the marginal bones, the peripheral bones here. And the ribs come out and meet them. In between the ribs, you'll see ossification. Okay. So you have the neural bones above the vertebra, you have the costal bones over here, and you have the peripheral bones. Over here is the nuchal bone, which is the most creative bone in the turtle. It differs greatly between species. The plastron, the 9 bones generally, with the 3 anterior plastron bones, the hyoplastron, the hypoplastron, and the zippiplastron being more proximal. Okay, so making a turtle. This is a picture from Louis Agassiz's book in the 1850s on turtle development. It's an amazing book. It's a beautifully illustrated book. It was illustrated by his friend Sanrel, who came from Switzerland to do the lithography. And where are the turtles coming? They're coming from Walden Pond from Henry David Thoreau. Henry David Thoreau, the philosopher, was collecting turtle eggs for his fellow transcendentalist Louis Agassiz. And Louis Agassiz did marvelous dissections of turtles, and this is Sanrel's depiction of a stage 14 turtle. And what you see here is, here's the head of the turtle, here's its yolk sac, here is the forelimb, here's a hind limb, here's the neck, and you can see the shell, and at the tip of the shell is this ridge. This is the carapacial ridge, and this is responsible for making the turtle a turtle. This carapacial ridge over here. Here is a stage 13 turtle, and you can see over here the ridge just beginning to form. Here is the notochord, here is the neural tube, here is this carapacial ridge, very rich mesoderm here, and I'm taking this slide through the limb bud. Here's the limb bud. Here's the AER of the limb bud. I don't want you to think that this is a limb bud, because this looks like an immature limb bud. And now, this is the carapacial ridge. This is still dorsal on the turtle. Here again, this is a few stages later. Here's the notochord, here's the spinal cord, and you can see the rib has come out, and the rib has gone through the myotome. Here are the cells that are going to produce the costal musculature. Note, it's not having anything to do with the myotome. It's going through it, and it's going into the carapacial ridge. It looks like it's being attracted to that ridge. And here's another picture. Here's the normal situation, chick, mouse, and humans. This is from a mouse embryo, where you could see the rib going ventrally. And here it is in a turtle embryo, where the rib is going eventually into this ridge over here. Okay, now, Chester Intima and Annie Burke looked to see if this really was an attraction. And so what Chester Intima did was he took, very carefully extirpated the carapacial ridge of turtle embryos. This is very difficult to do, and especially because the carapacial ridge wants to grow back. You have to do this really timed well. And so he took out the ridge here and asked, where did the ribs go? And what he found was the ribs went into ridges that were adjacent to the place where they should go. So instead of the rib going here, it went into another ridge, as if it were attracted to this ridge. What Annie Burke did was she put tantalum foil in between the ridge and the rib, so that signals could not get from one to another. A signal from the ridge would not reach the rib. And what happened there was amazing. The ribs went south. The ribs went ventrally instead of going, forward. And so this indicated there was an attraction of the ribs to the ridge. So we wanted to see what this attraction was. So Annie Burke had said, it looks like this is a limb bud. Maybe it's working the same ways. Maybe it's working through FGFs and WNTs, and maybe it's being attracted, like somite muscles are attracted by an FGF. So our lab decided, okay, let's look at